Hello everyone, I'm Afeefa Zainab and I will be talking about a research which is entitled as Importance of the Suitable Reference Data for Improved Cell Type Prediction in Spatial Transcriptomics Data. Spatial transcriptomics is a promising technique in providing cellular insights into tissue structure in the native states, which means that it provides a detailed transcriptional information in cellular context and helps in analyzing and visualizing the gene expression in native states. Although it's a very promising technique and widely used these days, it can have some of the challenges which I will be discussing in the next slide and which will be a focus of our problem. So, as I mentioned in the previous slide, the spatial transcriptomics is a very promising technique in measuring the gene activity in and allows the gene expression in the original physiological context, which is normally not possible by the single cell RNA. But one of the challenges that spatial transcriptomic data analysis faces is that prediction of the cell type at each spatial location. That can be one of the then can be because of the reason that the numerous cells or the mixed cell types can be present at each spot, which make it very hard to predict the accurate cell type composition. And therefore, this is one of the things that we really need to address. So normally what happens is that in order to predict the spatial, in order to predict the cell type in the spatial data, what we do is that we take a spatial sample and perform deconvolution method for which the single cell RNA seq reference is actually utilized, which is well annotated with the cell types. And it after performing the deconvolution, it actually predicts a cell type at the spatial location. For example, in this case, as we can see, for example, the fraction of medium spiny neuron is being shown in this tissue. But the problem comes here. So what happens if we do not have the suitable reference or the reference that we use is not suitable? Let's take an example. For example, we have a spatial data or the liver with the tumor, and then we are going to use a single cell RNA-seq reference, which is actually a liver data, but it does not have the tumor cells. So what it will do is that it will predict the cell type composition at each spot, but there will be no tumor cells predicted. So here, normally, if the reference would have been containing the tumor cells, then the prediction could be something like this: that there should have been clear tumor, which could have been predicted over here. But this is not the case because the reference did not have the tumor cells in the annotation. Another case, for example, is that what happens if we take the completely wrong reference. For example, in here, we took the spatial sample for liver and then we used a brain reference for the brain single cell RNA reference in order to perform deconvolution. And as a result, the predicted cell type composition was that it predicted neuron cells in the liver, which should not be there and which does not make sense because uh, so the point is that the problem happens when we choose the wrong reference or the reference that does not have the accurate cell type that it should be there for the deconvolution. So what should be done is that in order to pick the suitable reference, what we did is that we tried to create a suitable reference by manually selecting the cell types. For example, we chose the different cell types from the existing single cell RNA databases. And for example, we took the cancer cell types, tissue specific cell types, and immune cells, and tried to create an optimized single cell RNA reference containing all the possible cell types that should be there or should ideally be there in that reference, actually. So, based on this background information, we tried different approaches. For example, in the first case, we tried using a reference that was too general, which means it was having a diverse cell types for analyzing the breast cancer spatial samples. And this is the results that we obtained. So, in this heat map, as we can see on the y axis, we have spatial samples, and then the x axis are showing the cell types. And we can see the fraction of each of the cell type being predicted in these samples. And some of them I have highlighted to show the misinformation. For example, the basal cell of the prostate epithelium is actually being shown or predicted in the breast tissue. Although, and another strange one is the transit amplifying cell of colon, which is also showing quite a promising fraction in the breast tissues. Another problem is that there were no tumor cell predicted, although all the spatial samples that we used were actually from the breast cancer. So after trying the two general reference, we tried to use the reference that we created using a manually selected cell types, and these are the results. And in here, that really showed the promising result and a clear pattern. And most of the samples were with mainly the tumor tissue showed the higher fraction, and it could be seen over here. And on the other hand, it also showed that in some samples where there was some of the tumor tissue and also some of the healthy tissue, this is kind of clear pattern being shown in this map. So this actually showed the results that the cell type prediction was more accurate and true as compared to the one when we use the two general reference. So after seeing the results so far, we concluded that the suitable selecting or choosing a suitable single cell RNA reference is very crucial in order to predict the cell types with more accuracy for spatial data. And on the other hand, our approach also shows the promising results in accurately predicting the cell type composition as compared to the samples that were too general or contain some unnecessary cell types, or maybe in some cases lack the crucial cell types that should be there. On the other hand, although our approach, which is selecting the cell types manually, showed the promising results, but it was taking more effort. And if we want to apply it on the larger samples, I think it would be very time taking also. Therefore, currently we are working to automate this process so that it can be applied on more samples. And we are focusing on how to define and select the suitable cell types for each sample in the future. So if you have any questions or if you want to connect with us, kindly feel free to ask any questions. And the poster is also on the display. Please feel free to ask. Thank you for listening. Thank you so much.